Our next speaker is Carol Barnes, and she will, her talk will be about animal models of brain adaptation and compensation in aging. So if you were not paying attention to animal studies of compensation and resilience, you certainly will be after this talk. So please.
Thank you. Thanks, Naftali, and thanks for the invitation to be here. My role in this session is going to be to talk about parallel lines of work on brain and cognitive aging, resilience, and compensation in animal models of normative aging. Rodent and non-human primate models provide a number of advantages for studying aging, including the facts that, in fact, animals other than humans do not spontaneously show the most common human neurodegenerative disease, that's Alzheimer's disease, and physiological, anatomical, biochemical, and molecular circuits in the brain can be examined in greater detail in animals. But of course, the objective of our research is to guide understanding of human brain and cognition. So it may be fair to ask me at least 2 questions, and if I don't ask myself, I think Naftali will probably ask me. So I'll ask myself, why do I think that it's even remotely reasonable to believe that there may be a correspondence between brain and cognitive aging between species? And what have we learned about any of the concepts that this summit is focusing on today from animal models? Well, to partially and quickly answer the first question, I'll focus on the hippocampus, a phylogenetically ancient structure that's well conserved across animals and critical for certain types of memory in all species who have been tested. Even turtles have a lovely primordial hippocampus, or archy cortex. So this slide shows what was portrayed to be true about cell loss in human temporal lobe when I first became interested in aging in the early 1970s. By the time you are 90, your interminal cortical cells would be very shriveled. But over the decades, with well-controlled humans studies, non-human primates, and rodent experiments, one thing we know about the hippocampus during aging is that widespread cell death is not the norm. In fact, the numbers of principal cells, such as granule cells, CA3 and CA1 pyramidal cells in the hippocampus, do not decline spontaneously in animals at ages nearing their species-specific lifespan. So not in humans, not in monkeys, not in dogs, rats, or mice in which these cells have been counted with blind experimental designs and stereological methods. But we know that in each of these species, there are memory deficits that suggest some dysfunction of internal and external hippocampal circuits. Now, spatial memory is more straightforward than some other kinds of memory to examine across species because it does not require language to test its functionality. And spatial memory is observed to change at 2 years in rats, 22-year-old monkeys, and 65-year-old humans. Each of these ages considered to be old in these respective species. However, I think we need to change the definition of old in humans because I'm now well over on the other side of 65, so I think we need to discuss that, but that's for some other time. In this example from rats, the data is shown from Mark Lindner's lab. Shorter path lengths of animals swimming in the Morris Water maze task is thought to reflect more accurate navigation and spatial memory, and older rats are worse on average than their younger counterparts. To the right, you see data from Scott Moffat's lab. Shorter path lengths in a virtual reality navigation experiment and environment also reflects better memory of the environment, and this also changes as a function of age. Older people are also worse. But what you notice from these data is that in both rats and human data, there's some variability with older rats and people showing quite good memory, and some others quite poor. So as I said previously, hippocampal cell numbers don't change with age across species. So why is there a change in memory, and what's responsible for the variability and the degree of memory change? Now this cognitive variability itself suggests individual differences in the extent of brain adaptation in aging. Now, from animal work, we have several clues about where to look for these brain changes. First, we know that one form of synaptic plasticity that we believe is used by the brain to lay down memory traces, long-term potentiation, or LTP, is altered in the aging hippocampus. Now, while synaptic strengthening can be induced at old synapses, and by inference, these aging synapses can store information, we've shown that the strengthening decays more quickly in older animals, and this faster decay is correlated with how good the rat's memory is, actually regardless of age. Furthermore, another kind of plasticity that reduces synaptic strength, long-term depression, or LTD, is easier to induce in aging hippocampus, which was shown by Tom Foster's lab. So from animal work, one answer to the question of why memory changes with age is that these plasticity mechanisms are altered. Another thing we know from animal model work is that while cell numbers remain reasonably stable, synapses, these important points of communication between cells that wire up critical memory circuits in the brain, synapses are lost during aging. For example, early work by Geinesman and his colleagues and electrophysiological data from the rat suggest axon pruning and a loss of up to about a third of synapses from the medial entorhinal cortical projection into the hippocampus. These findings have been replicated many times in animal models with a number of newer techniques. And in fact, more recently, this kind of white matter pruning has been confirmed in healthy older humans, including work from Craig Stark and Leila de Toledo-Morel's labs, where using MRI methods, their data suggest a thinning of the white matter tract connecting the entorhinal cortex and the hippocampus. So we have evidence then from animal models that suggest that synaptic loss also contributes to memory decline, and presumably the degree of change in these plasticity mechanisms and loss of connections could in part explain differences in cognitive reserve and resilience between individual animals. Now, I want to say a little bit more now about how animal models have contributed to conceptualizing ideas about brain compensation in aging. I'll start with this ancient text published in 1980. I suppose you could call it classic literature to be less judgmental, but the data in this manuscript combined in vivo electrophysiological recordings of synaptic potentials in young and old rats that I'd collected for my thesis experiments at Dalhousie University, working in Graham Goddard's lab. And because the electrophysiological measurements that I obtained were extracellular, there was some or several alternative explanations for reduced amplitudes of the synaptic and presynaptic responses in the dentate gyrus of old rats. An exact interpretation could only be given by doing intracellular recordings so that I could get direct voltage measurements. So in the late 1970s, the best place to learn in vitro hippocampal slice technology was Per Anderson's lab in Oslo, and Per said he would give me a spot in his lab if I could get a fellowship. I'm not sure why I thought the US government would support somebody who had been in Canada for 7 years for graduate school to then go off to Norway for 2 more years. But I applied in 1978 for a postdoctoral fellowship in NRSA from the National Institute on Aging, which had just been established in 1976. And Zaven Khachaturian, who was the neuroscience program officer, or whatever they were called in those days, took a bet on me. And I got the very first NRSA in neuroscience from the National Institute on Aging and went off to Norway in 1979. Bruce McNaughton was able also to get an insert postdoctoral fellowship from Canada to do other work in Anderson's lab. So in Oslo, we obtained evidence consistent with the idea of a pruning of axon collaterals and synapses from the medial entorhinal cortical projection onto hippocampal granule cells in old rats in the in vitro hippocampal slice preparation And these data replicated the in vivo recordings that I had done in Halifax. Now, the fiber potential is a reflection of the number of axon collaterals from layer 2 of the entorhinal cortex that can be activated. And you see here there's no change in the activation threshold, but evidence for a reduced number of presynaptic fibers in old rats. And to the right, even though the fiber potential response was smaller in the older rats, the synaptic responses were proportionally greater in these old animals. And it was critical that this finding held both for extracellular and intracellular voltage measurements, suggesting that the remaining synapses were actually slightly stronger in old rats. Now, this result was consistent with the idea of compensation at a single synapse level. And the plausibility of this interpretation was strengthened when, in 1991, Tom Foster conducted what I can only call heroic quantum analysis experiments in our lab using minimal stimulation techniques. With this method, you can stimulate a single axon collateral and measure the depolarization of granule cells generated by a single synapse. So Tom was able to then directly show that the strength of individual synapses was increased in old rats. With respect to the idea that the aged brain might actually actively adapt in response to a primary change in the number of synaptic contacts, the compensation narrative went something like this. Okay, if old granule cells can lose up to a third of their synapses from the medial entorhinal cortex, and we know that each granule cell needs a certain level of depolarization or activation to fire an action potential, then how do these cells transmit incoming information to circuits downstream of the hippocampus? And the idea emerged at that time that, well, the hippocampus may solve this reduced input problem by increasing the strength of the individual synapses that remain on old granule cells. And this is how the compensation was defined in these early experiments. But the question is, how do we know this is compensation and that synapse loss is the primary trigger for synaptic strengthening? And here you see a schematic of fewer but stronger synapses in old rats onto granule cells. And stronger synapses in old animals may well help counterbalance the effect of synaptic loss. But Monica Chawla in our lab has shown that fewer old granule cells actually are activated during behavior by using gene expression activity markers to monitor cell spiking. So this suggests that the increase in synaptic strength is not sufficient to completely restore normal network activity during behavior, so that this compensation doesn't mean that the circuit is back to normal completely. But the open question is then, how then would the network behave if there was no synaptic strengthening? The argument made is that the system might fall below some critical threshold for functional circuit throughput. And this hypothesis directly predicts that aged animals with higher cognitive ability may have more synapses, stronger synapses, or both than those animals showing low performance levels. Now, the experiments to test this hypothesis have not been done, but the results could either refute or support the compensation idea. Always good to have an experiment that could actually disprove your hypothesis. But if this was positive, this would point to changes in synapses as being a primary trigger for adaptive responses in aging brains. One important thing to keep in mind, and I know Cheryl Grady and others will have more to say about this, you can't just look at one part of the circuit to understand the aging brain vulnerability or adaptability. Rather, you need to keep in mind the complexity of the circuit examined and how perturbation of one component of the network will likely impact the regions to which it's connected. And I'm focusing here on the input from the entorhinal cortex to the hippocampus for simplicity. And even this myopic view, you can see, can become quite complex. So I'm going to give you an example now, my final example, C3 pyramidal cells. There's a reduced input from the dentate gyrus. I just told you that fewer granule cells are active during behavior. Peter Rapp's lab has shown that CA3 also receives reduced input from the entorhinal cortex, but from these facts you might have expected that CA3 cells would be less active. But we know Ian Wilson with Michaela Gallagher showed that CA3 pyramidal cells in aging rats show higher firing rates than young. Alex Tomei in my lab has shown that CA3 pyramidal cells in old monkeys show higher firing rates than young. And Mike Yassa has shown that older adults show increased bold signal in CA1 region of the in CA3 region of the hippocampus. So could part of the explanation for this hyperexcitability be the result of a compensatory strengthening of synapses from CA3 from the dentate gyrus and entorhinal cortex, just like we saw for the granule cells? Again, experiments to explore these ideas have not yet been done. Another change in CA3 circuit that could help contribute to this hyperexcitability is a change in interneurons. So while it remains controversial whether a decrease in inhibitory neurons is observed in CA3 in rats, in monkeys, Alex Tomei has shown that there's a reduction in somatostatin neurons associated with higher firing rates in CA3. I'm running out of time, so I won't go through the details of this, but in fact, the neurons, the fewer neurons is associated with greater firing rates. So what do these, how does this change happen? We think it's a change in chemical phenotype in these interneurons because the same numbers of neurons overall exist. But what do these data imply? Well, could there be two compensatory responses to a decreased drive onto CA3 from the dentate gyrus and entorhinal cortex? Increased synaptic strength and decreased numbers of inhibitory cells. Is this compensation gone wrong? I have several thoughts to leave you with. One provocative issue I raise in closing is that if our goal is to assist the aging brain to achieve an optimal state or function at older ages, I think understanding the compensatory or adaptive reactions to primary age-related changes is going to be critical. Another point to raise is that the use of non-human primates is going to be important to bridge a gap between rodent and human aging, and this resource is under great strain right now, and I'm hoping it will survive. And finally, we need to fully embrace the idea that the brain is not passively sitting back during the aging process. The brain is continuously adapting and changing throughout the lifespan, making it crucial to be strategic in our attempts to modify the circuits within this really magnificent and actively adapting organ. And just quickly to say I'm grateful to the McKnight Brain Research Foundation and NIA for all the years of support. Thanks.
We have time for a couple of questions, so please step to the microphone.
When do you think these changes begin? Most of the work you're talking about, particularly with the hyperexcitability, is in the very old. So we're looking at adaptation. What should we look for earlier?
Right. So most of us don't do longitudinal studies in our animals because you have to sacrifice them at the end and so forth, and the implants don't last that long. And we can't precisely answer the question of when it starts. My intuition is that we need to go earlier and earlier, not only, if we're gonna alter cognitive trajectories over the lifespan, we need to get those manipulations in the system as early as possible so that we can optimize how we will go forward in aging. So I don't have a pinpoint answer to that now. Catherine.
Yeah, great talk, Carol. My question is similar to Tom's. What do we think the initiating events are that we need to compensate for? And I think part of that will be longitudinal studies, but is this going to be, are we looking for a molecular switch, a cellular change, or a network phenomenon?
It's going to be cells, but how they participate in the network, it's got to be. So I can't separate those. I think there's a number of molecular switches that might be modifiable to keep our circuits more plastic and to evolve in a more favorable way. And we're all trying cognitive interventions, dietary interventions, lifestyle, and so forth, to try to see what the maximum effects would be by doing those kinds of manipulations in aging. Hi.
Hi, fascinating talk. Dogs kept in people's homes who live well beyond their natural lifespan, of course, do develop beta amyloid 1 to 42, and veterinarians see dementia very often in dogs that are 12 years old and so on. Are you quite sure that your rats, I haven't heard anything about rodents and beta amyloid, don't have beta amyloid?
It's still dogs do not develop the pure classical symptoms of Alzheimer's disease. They don't get hyperphosphorylated tau and so forth. And cerebral angiopathy is one thing, these plaques are another. So no, rats don't. And I have a study ongoing very carefully characterizing monkeys over the lifespan that I hope to get out this summer to show that they also do show some plaques, don't show tangles, and their cognition does change. So.
I'm sorry, I think we're out of time, so I'm sorry, but we'll have to save this question for the general discussion.